13. Okay, next up is a report from Dr. Brian Lapointe on the Indian River Lagoon as a bioreactor, new perspectives on sewage pollution. And I believe, is this the report that was given to RAC?
Yes, sir.
Yeah, and it was very well received. I was told it was very informative, and so we asked to have it brought in front of the entire board. And here to present is Dr. Brian Lapointe.
Great. Good morning, and thank you, board members, for the invitation to present this today. I do also want to thank Laura Herron, my assistant in this work. She's been with my program for about 5 years. And I think, as we all know, the Indian River Lagoon has faced some very critical issues, particularly in the past 5 years. We've heard, of course, a lot about the excessive freshwater releases from Lake Okeechobee and other sources. Nutrient problems coming from not just from fertilizers, but as we're going to talk about today, I'm going to look at other sources of nutrients. These can come, obviously, from a variety of sources, including sewage, which also brings microbial pathogens, viruses and bacteria, and other contaminants as well. The nutrients, of course, are linked to the harmful algal blooms that we're seeing in the lagoon. These blooms can be comprised either of macroalgae, large seaweeds that grow on the bottom. We see some photos on the bottom showing those, but also phytoplankton, single-celled algae that bloom, like the microcystis, the toxic cyanobacteria, the green photo on the right, and also the brown tide, another phytoplankton that basically blocks light to the seagrasses and causes loss of seagrass habitat. That, of course, no habitat, no fish, and it affects the fisheries. And what we've seen over the past 5 years, particularly in 2013, were emerging diseases and mortalities in wildlife, including manatee die-offs, dolphins, sea turtles, pelicans, fish, and shellfish. Well, we've been studying nutrients at Harbor Branch a long time. In fact, that's me about 40 years ago when I first came to Harbor Branch as an employee, actually of Woods Hole Oceanographic Institution at the time. And we were involved in this visionary research to try to basically perform advanced waste treatment to sewage effluent by passing it through marine aquaculture systems. These were open linear systems that included phytoplankton ponds, shellfish raceways, fish, and macroalgae, these seaweeds. They're like sponges for nutrients. They were grown at the very end of the system. And that particular plant I'm holding there is Gracilaria. It's a red seaweed that grows very rapidly in the presence of high nutrients. It grows very well in seawater sewage mixtures. And we demonstrated in that work that actually that plant, Gracilaria, has a very high capacity to remove nutrients in these flowing systems. Well, since we did that work in the 70s, we've seen these seaweeds and other harmful algal blooms expand in South Florida's coastal waters. We've studied these problems in the Florida Keys, Lee County, there in, 2003, 2004, had big problems with these red drift algae. A lot of that was that Gracilaria plant, and the Indian River Lagoon as well, where these overgrow and smother the seagrasses. So the problem comes down to nutrients, and where are the nutrients coming from? Well, as it pertains to the Indian River Lagoon, what we've seen over the decades is actually increasing urbanization of the lagoon. We see up to about 40% of the watershed is now urbanized. Agriculture, actually, in many parts of the watershed has actually decreased, but still a sizable area is used for agriculture. And in 2007, scientists from around the nation, we came together to look at our nation's estuaries and look at them very carefully with respect to this nutrient and eutrophication problem. And we knew then that the Indian River Lagoon had a moderate to high nitrogen input, and it had a very high susceptibility for eutrophication, the buildup of nutrients, and these harmful algal blooms, particularly in the northern part of the Indian River Lagoon in the jurisdiction of the St. John's River Water Management District, between Ponce Inlet and, in this case, down to Sebastian Inlet, where there's very little flushing of the lagoon. From Sebastian Inlet south, we have a greater amount of flushing, particularly between Fort Pierce and Jupiter Inlet. But we knew also that at that time, the Indian River Lagoon already had a substantial expression of eutrophy from seagrass loss and algal blooms, and we knew that algal blooms were likely to worsen. And indeed, that's what happened. As nutrients build up in these systems like the Indian River Lagoon, initially you can see a positive response of the seagrasses. Like other living things, seagrasses need nutrients, but at some point the nutrients become excessive. You begin to see that kind of continuum of photos on the bottom going from clean, productive seagrasses that get increasingly overgrown by first epiphytic algae that overgrow the blades, then the larger macroalgae, and ultimately at very high nutrient loads, the phytoplankton come in, and that's, of course, when you really lose the light and you go into kind of a rapid seagrass decline. And that's exactly what happened in 2011 when we saw the super bloom develop for the first time ever in the Indian River Lagoon. You can see the slow increase in seagrass cover in that graph from 1986 up to 2010 or so, and the precipitous decline after that. We had a brown tide in 2012. Both these were unprecedented phytoplankton blooms, which of course resulted in unprecedented seagrass die-off, and was followed by the wildlife fish and shellfish mortality in the lagoon. So we've heard a lot about various sources of nutrients, particularly fertilizers, both agricultural fertilizers and residential fertilizers, and their role in supporting these algal blooms that we're seeing. And we've had ordinances passed since this problem developed to deal with residential use of fertilizers. But no one was really talking about sewage and what it was contributing to these problems in the lagoon. And if you look back at the Indian River Lagoon Act of 1990, it did a pretty good job of eliminating the sewage outfalls into the lagoon, the point sources. And it also had language pertaining to the septic tanks, because the architects of the act, and many knew there was a septic tank problem. These problems were seen in the 1980s. But the language in the act pertaining to identification of hotspots of septic tanks and septic to sewer conversions really didn't go anywhere. And today there are on the order of about 300,000 known septic tanks on the Indian River Lagoon watershed. Some estimates go as high as 600,000. No one really knows exactly how many there are. The soils in many of these areas are unsuitable for septic tanks. They're sandy, porous soils, very low carbon content. And the contaminants in the septic tank effluent include nitrogen, phosphorus, organic wastewater compounds, things like pharmaceuticals, hormones, and pathogens, bacteria and viruses. And if you just do the math, how much nitrogen can come from these septic tanks, just 2.5 people on average per septic tank. The estimated nitrogen load up and down the lagoon is on the order of 2,575 tons of nitrogen per year. Now, to put that in context, I think your water report for the Lake Okeechobee total nitrogen load coming east is about 520.8 tons. So it's substantial load. In fact, recent studies by Bedrusman show that septic tanks are now considered the second single largest source of nitrogen in Florida's surface waters, behind fertilizers. And there are other issues with septic tanks, how they can alter the biogeochemistry of the groundwater and the surface waters. Research up in Cape Cod by some of my Woods Hole colleagues have shown that sulfur-reducing bacteria in these anoxic plumes in the groundwater can methylate mercury, making it bioavailable. And we do have very, very high mercury concentrations in the fish and dolphins in the lagoon, as well as the humans that are eating the fish coming out of the lagoon. So, I actually got started studying septic tanks a long time ago in the Florida Keys. The second study I did was in 1995, getting up closer to the Indian River Lagoon. This was in collaboration with the Loxahatchee River District, where we studied Jupiter Creek estates, shown on the left. There were hundreds of septic tanks that were flowing into Jupiter Creek that flows down in the southwest fork of the Loxahatchee River. And I worked very closely with Steve Krupa, hydrogeologist here at the district. I had a groundwater flow meter. This, I think, was the first groundwater flow meter in the state of Florida. You also have one now, I believe, called the Krupa Seep. And this is where it started, right here, with this study. And it was a wonderful collaboration with Steve Krupa. We looked in great detail at the interaction of the groundwaters being impacted by the septic tanks and Jupiter Creek and the tidal pumping effect and how the groundwater can accelerate as the tide ebbs and falls, bringing those contaminant plumes into the surface waters of the southwest fork of the Loxahatchee River. We also saw for the first time how failing septic tanks, and I put that in quotes because septic tanks are not engineered to remove nutrients or pathogens. So, but in this sense, this goes to the DOH definition of a failing septic tank being less than 2 feet from the water table. And in this case, what happens, the waste effluent from the septic tanks doesn't have adequate vadose zone to nitrify the ammonia to nitrate. And we get ammonium buildup in the groundwater, which limits the amount of denitrification or removal of nitrogen that we could get naturally as that waste plume travels towards the surface waters. We also, for the first time, used stable nitrogen isotopes to actually fingerprint the source of the nitrogen. There are many sources of nitrogen, but it turns out that wastewater nitrogen has very high nitrogen isotope values, and we can separate that from fertilizer nitrogen or other natural nitrogen sources. And we saw very clearly in this study that the shallow groundwaters affected by the septic tanks and the creek waters had very high values of these nitrogen isotopes, clearly showing this is coming from human sewage. And we also saw other evidence, high fecal coliforms in the wet season in the groundwaters and the surface waters, where the infiltration by the rainfall would push, these coliform bacteria right through the soils into the groundwater, and they would get transported into the creek. And we also looked at caprostanol, a fecal sterol, another tracer of human fecal input that builds up in the sediments. Well, that study was followed by a study about 10 years later in collaboration with the city of Estuary and Martin County. And if you recall, 2005, when we started this, a very, very wet year, record hurricane activity. And we began sampling in June and November of 2005 during a time when we did have a lot of water releases from Lake Okeechobee. And then also in March 2006, when the salinity came up, there were lower discharges. And so we clearly saw the effect of freshwater discharges, and again, not just from the C44, but from the C23 and C24 as well. We did see the highest turbidity nitrate, total nitrogen, in the South Fork. Interestingly, in the North Fork, very high concentrations of ammonium and phosphate. Okay, and there are a lot of septic tanks as we go up towards the Port St. Lucie area on the watersheds of the North Fork. The highest nutrients and coliforms we found near residential areas with high densities of septic tanks. And this is the first time we saw the microcystis blooms in the estuary. We got an opportunity to resample those cyanobacterial blooms in 2013. where we again looked at the isotopic signature, and it was quite high, 8.6 parts per mil, which puts it in the range of wastewater nitrogen. So although these toxic cyanobacteria can come from freshwater sources, either Lake O or others coming down the various canals, once they get in the estuary, they can respond to local nutrient sources, both phosphorus and nitrogen contributions from septic tanks. That can enhance the bloom formation. And that brings us up to the next study, which was we took these methods that we've been using in the previous two studies, and we went Indian River Lagoon wide. All the way from Ponce Inlet up here in the Mosquito Lagoon in the north end, south to the Jupiter Inlet. We had 20 sites and several reference sites outside the lagoon. And the objectives was to use multiple lines of evidence, nutrients, carbon, nitrogen, phosphorus levels in algae, and the isotope values in macroalgae to assess the patterns in nutrient pollution and algal growth as they're responding to various nutrient sources up and down the lagoon. And our goal was to improve water quality in the IRL by providing high-quality, user-friendly data to resource managers and policymakers. And you can see the map. The segments that we looked at, of course, are the Mosquito Lagoon in the far north, the northern IRL, the Banana River, the central Indian River Lagoon, and the southern IRL. So when we look at the data from these 5 segments of the Indian River Lagoon, and we see the reference site on the far right, you see a very clear pattern in nitrogen. This is total dissolved nitrogen, and you can see the southern IRL near the reference site, slightly elevated above the reference site, but as you go north, you can see those concentrations during the 3 samplings we performed starting in the dry season of 2011, and that was at the end of that prolonged drought, to the wet season in 2011 and the wet season in 2012. You can see how that nitrogen just goes up, 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 peaking in the Banana River, which is where we saw the initial super bloom form. And it goes down somewhat into the Mosquito Lagoon. And you can see clearly the IRL target of about 50 micromolar. You can see how we're really at or above that from the central Indian River Lagoon going north. Total dissolved phosphorus was a very different pattern. We see actually the highest concentrations in the central Indian River Lagoon. This is coming with fresh water. During the study, we had the lowest salinities in the central Indian River Lagoon. The St. Sebastian River was a major source of that fresh water, as were the relief canals in Indian River County. So if you do the kind of the math or calculations in your head, with the high nitrogen in the northern IRL and Banana River and the lower phosphorus, you can see how it has very high nitrogen to phosphorus ratios up in that part of the lagoon that we think might be linked to the toxicity in some of the algal blooms that we've seen up there. The unique part of this study, others have looked at nutrient patterns in the Indian River Lagoon, but no one had ever done this particular type of research before, where you take these algae blooms, in this case the macroalgae or seaweeds, and use them as little bioobservatories. These algae are assimilating nitrogen 24/7, and by collecting them and analyzing them for the isotopes, you can make some inferences as to the source of the nitrogen causing these harmful algal blooms. And if you look at that little table on the upper right, you see the isotope level, the N15 level, and you can see that septic tank effluent has values of about +3 to +5 parts per mil coming out of the tank. And whether it goes through the ground or, say, to a wastewater treatment plant, the more processing of that effluent, the higher that value goes. So we tend to think that wastewater values have values of +3 parts per mil or higher. Now, below that, you can see that all these other sources, upwelling of cold North Atlantic water has high nitrate. We have nitrogen fixation, a natural nitrogen source, atmospheric nitrogen, fertilizers, and Everglades peat all have lower values. So by collecting these algae, analyzing them, it can tell us what the source of the nitrogen is. So if you look at the panel upper left, the data from the Indian River Lagoon, all the segments, you see very clearly that these values are quite high. They average about 6.3 parts per mil lagoon-wide. And if you look at the bottom panel, the yellow box is the Indian River Lagoon. It's neck and neck with the same type of study that was done in Boston Harbor when all the sewage was going into Boston Harbor prior to the diversion. So when we saw this number, we knew we had a wastewater problem in the Indian River Lagoon that had not been recognized. And on the other hand, if you come down on that bottom panel to the very right end, you see that low value Western Florida Bay. That's reflecting other sources of nitrogen. Could be Everglades peat, atmospheric nitrogen, fertilizers. All those would bring that value down to a very different signature than we're seeing in the Indian River Lagoon. This is a paper we published about a year ago. If you would like this, we can get a copy of this to you. It's got a lot of information about this study and about the wastewater issue in the Indian River Lagoon, published in the journal Harmful Algae. Well, we took it one step further. During that study, we realized that we should really look more closely between the lagoon, the main stem of the lagoon, and the interface, these tidal creeks, which are lined by septic tanks in Indian River County. And one of my students, Marie Tarnowski, did this work for her master's thesis at FAU. And what she did, she really built on the study we did with the Loxahatchee River District, sampling the relief canals, the North Relief Canal, the Main Relief Canal, South Relief Canal in Vero Beach, as well as along the St. Sebastian River, both in areas of high densities of septic tanks and more naturally vegetated reference sites. We looked at surface water, groundwater. So I'm going to just breeze through a little bit of her results. She defended this a year ago, December. When you pool the data into natural versus residential areas, you see ammonium, the dissolved inorganic nitrogen, total dissolved nitrogen, soluble reactive phosphorus, and total dissolved phosphorus are all higher in these residential areas that rely on septic tanks. Wasn't really too surprising. Now, we also looked at the plants for the nitrogen isotope, and what we see again, the bars on the very left, the St. Sebastian River, you can see those values are roughly 6 to 7 to 8, very close to our average values for the Indian River Lagoon, and that would be expected. Major river and water source flowing into the lagoon. But look at the relief canals. They're significantly higher than the river. Values up around 10 up to 12 parts per mil for the isotope values. These are values we typically see at the end of treated sewage outfalls. So these canals are really functioning like wastewater outfalls coming in to the Indian River Lagoon. And if you look at the dry season, the red bars are slightly higher than the wet season, and we attribute that to, again, rainfall in the wet season bring lower isotopic signature of rainfall itself and fertilizers, residential fertilizers, into the system that would bring that value down a little bit. Well, you know, the public can't follow the nitrogen isotope stuff very easily, so we wanted to get another tracer that they could really relate to. So we also looked at sucralose, the artificial sweetener that is commonly used, and we analyzed that in these canals and actually compared it with the nitrogen isotope values. This is Marie on the bottom left panel. And the thing about sucralose is it goes right through sewage treatment plants. It's a conservative tracer of human wastewater. And that bottom graph showing the blue bars just shows the very high concentrations in wastewater effluent, and that as you come down near septic tanks, surface waters are often have these higher concentrations of sucralose that dilute down to very low concentrations when you get a lot of dilution, showing the clean value over on the right. So we see a very high correlation of sucralose with total dissolved nitrogen, that one graph on the left, and also with N15, okay, the graph on the upper right, showing that as sucralose concentration goes up, the N15 goes up, again, both pointing to a human source of the nitrogen enrichment we're seeing. Well, as I brought this information out in talks, and our paper was published a year ago, you know, I've gotten a lot of feedback from engineers saying, you know, we've always known this, you know, septic tanks aren't designed to remove nutrients and pathogens, but I think the public doesn't really understand that. It's out of sight, out of mind. They think it's gone away once it's underground. The reality is septic tanks are not protecting the Indian River Lagoon or other resources where you have high densities of septic tanks in particular. One septic tank by itself isn't a problem, but when you build out these urban areas with thousands and thousands of septic tanks, it becomes a problem. It's an inadequate infrastructure problem on the watersheds of the Indian River Lagoon. It was identified as a problem in 1990 in the Indian River Lagoon Act, but not acted upon. So we're kind of 20 years behind on the Indian River Lagoon. We really need to go back to where we were back in the early 1990s and pick up from there. And we do see increasingly with different counties on the Indian River Lagoon, Martin County, Volusia County, are making steps to do septic to sewer conversions. And increasingly, of course, they're able to get credit, nitrogen credit, by removing these septic tanks and taking them offline in their BMAT programs. And with that, I think I just want to acknowledge this kind of work takes a village. And we've got a lot of great help these days at Harbor Branch because of our FAU collaboration with students. We've had a great team to help get this work done. With that, I'm going to wrap it up, and if you'd like to ask any questions, I'd be glad to answer.
Thank you, Dr. Lapointe. Any board questions or comments for Dr. Lapointe regarding this presentation? Mr. Powers?
I just have a comment. Thank you very much for the presentation. It's very informative. It validates what most of us know, and that the problems are multidimensional, but without the science that backs that up, you don't know what those solutions need to be. And this is a key element in identifying what has to be done for solutions moving forward. So very helpful, and thank you very much.
Great. Thank you.
Mitch.
I agree with that. I think that this is important work that you've obviously been working on for 20 years. You've got to have the patience of Job, because it seems like you've been saying the same thing for 20 years, and just now folks are beginning to pay attention and recognize the importance and the significance of this issue. And, you know, I know I've been in lots of conversations over the last at least 10 years where, you know, there's been continued misdirection about what the cause is. It's not to say it's one fault or the other fault, but that they contribute together, and you've got to address both of these issues. So, A, thanks for your diligence, and B, thanks for the work, because I do think that this provides direction on how do we move forward. One issue that I wanted to clarify, and you touched on it briefly on the end, was the purpose of septic tanks is not to remove the nutrients. But I've also talked with folks that will seem like they're willing to say all septic tanks are bad. And I just want to get some feel from you. Does the density of septic tanks, the soils where septic tanks are located, the depth to the water table, the distance to a natural water body, do those have an impact or a role in the treatment cycle from septic tank to water body?
Yeah, well, it is complex, but, you know, we were guided early on by a wonderful report done for at that time, the Department of Health and Rehabilitative Services, HRS, now DOH, that permits the septic tanks. And this report, the Bickey report, and we cite it in our paper, back in 1982, I think, when it was done, pointed out the complexity, the biogeochemistry of the nutrients coming out of the septic tank, but that all that research that was done really in the 60s and 70s on septic tanks showed that there are these very high concentrations of ammonia and the complexity with the soils, the reactions with the soils in terms of treatment. And you need high amount of organic matter in the soils to get what we call denitrification. And in many of the soils in the Indian River Lagoon area, we don't have that. You know, these are very sandy soil, very porous also, that allows rapid migration. Another thing I think you've heard a lot of people talk about, some people have claimed plumes don't move more than 50 feet. So if you're more than 50 feet or 100 feet from a water body, then it doesn't get there. But again, the more research that's done, mostly by hydrogeologists, these plumes move a long distance, miles, way more. And so this is part of the confusion, I think, that the public have, that, oh, you know, these things just go and maybe they move a little bit this much a year. I'll tell you, there's a wonderful short video that was produced by the city of Tallahassee, TAP, Taking Action for Personal Pollution. And you can actually just Google it on YouTube. Go to YouTube and punch that in. And it's the city of Tallahassee talking about the problem at Wakulla Springs, a natural treasure of Florida, and how the spray irrigation field for the city's wastewater treatment plant, many miles away, I think it's 20 miles away, 22 miles away, that in combination with thousands of septic tanks are the nitrogen source that has created these very serious green algae blooms in Wakulla Springs. And it's a very well done educational film, but it just shows you how rapidly these problems can happen. That groundwater fluorescein dye test by Dr. Todd Kincaid showed that this water can move up to a mile a day.
Okay, Melanie, I think you had a question or comment?
Yeah, I want to thank you for coming to the governing board. I became aware of this issue when I was reading the UF study, and you talked about public awareness. I had no idea that this was going on, and so I reached out to you. You came and met with me, and then Mr. Moran was kind enough to bring you to RAC, and that was really amazing. So to have you here today is really special, and I appreciate you bringing this to the public's attention. You know, I think that it really shows that with an effort at a local level, you know, every person can make a difference. This is something that can be rectified. I know when I read this data and the first time I ever saw this presentation, it was really shocking. And I think that everyone that's seen it has had the same reaction. And I really applaud you for your work and your diligence. And I hope that through continually to get this information out, that we're starting to make people aware that they can make a difference in their own backyard. You know, years ago, and when we see it today even, we have a lot of people that live on the rivers that are very passionate about the health on the rivers. But at the same time, those same people who have been living on the river and caring about the river have been flushing their toilets for 30 plus years. And that is a contributor. And I think that looking at it in the scientific way that we are through your presentation here, but also working collaboratively, agency-wide, local initiatives, you know, there is a solution to this. This isn't an open-ended issue that we just keep talking about. This is something that is going to start to get better and better and better. And it's my understanding that your presentation to the counties up there has really yielded a lot of forward progress. So I appreciate everything you're doing and everyone involved with your team. It's a fantastic awareness on an issue that's really severe, and hopefully within the next few years we start to see a major recovery from humans taking responsibility and mitigating their own waste.
Mr. Moran.
Likewise, Dr. Lapointe, I want to thank you for coming, and thank you again for making this presentation to RAC and making us all aware of how complex these problems are. And I just wondered if you could estimate percentage-wise if we are fortunate enough to moving forward to have most of these septic tanks converted into, for these properties to get off septic tanks, what percentage of the nutrients going into the lagoon would be eliminated?
Well, the total load, that number was, I think, 2,575 tons of nitrogen. So, I mean, if we sewered everything, that would be the load that would be removed. And I can tell you from what's going on in the Florida Keys, that's where I did my first septic tank study. And, of course, these septic to sewer conversions don't happen overnight. That has taken 20 years. Between the time the decision was made by the governor and cabinet back around 1996, here we are 2016, and it's not quite done yet. The Lower Keys, the Cudjoe Regional Plant is going to go online within the next month or two. I have a home in the Florida Keys, in the Lower Keys, and I'm glad it's finally happening. I wish I had it sooner, but this was the headlines of the local paper over the holidays, the news barometer in the Lower Keys. Effluent was top topic in 2015. It was pretty much in the news every week, talking about how great it is that the sewers have finally arrived. And where they have already arrived, in Key Largo, they're almost 100% on sewer. And most of Marathon is sewered. The reports we're hearing from people that live on the canals is that this is a miracle, the improvement in water quality that they're seeing.
Ms. Batchelor.
Just briefly, Dr. Lapointe, thank you for your presentation. I also was clueless, and it was very interesting and quite sobering. But just briefly, I think your report, your findings underscore how critically important it is to have sound, objective science when crafting environmental solutions and policies. Because once you have the science, it really provides a path forward. So again, thank you very much.
Any other questions or comments for Dr. Lapointe?
Just an add-on anecdotal to your what's happening in the Keys over the holidays. My cousin was up from Largo, and this is a guy that grew up as a river rat on the St. Lucie, and he now makes his living in the water and on the water down in the Keys. And I asked him, I said, now that they're 100% converted, he said it's like a miracle. He said it's unbelievable. Within 30 days, he noticed, and he saw species that were coming in. He's on a little lagoon there. He said, Kevin, there's species coming in there we didn't even know existed. It's really flourishing. It was incredible.
Yeah, I'm looking forward to seeing that in my canal. All our beautiful turtle grass, I watched it die off over the years as more and more septic tanks went in to our subdivision. So, yeah, we're expecting to see some really good days ahead in the Keys.
Thank you very much, Dr. Lapointe.
Thank you.
Thank you. Okay, that brings us to agenda item 25, and we'll call on Jeff Summers.